on our ABIF YouTube channel. And I'm happy to share the slides if anybody's interested as well, because I think one of the goals is if people are interested to get involved with these networks. They're all community-driven and volunteer-driven. So the groups I'm going to talk about are the Canadian Network of Scientific Platforms, which is a broad network that focuses on not just bioimaging, but all technology platforms across Canada. So anybody who's running a research infrastructure platform and offering it as a service to the community. Canada BioImaging, which is part of the Canadian Network of Scientific Platforms and is really focused on the bioimaging needs of the community. BioImaging North America, of which Canada BioImaging is part of, and Global BioImaging, which was actually where things started for me before we formed Canada BioImaging and BioImaging North America. And lastly, I'll talk a little bit about core Apolini. So I was not involved in the naming. I think I love the acronym, but the long name is very long. So it's Quality Assessment and Reproducibility for Instruments and Images in Light Microscopy. And it's really an international group that's interested in improving the quality management of light microscopy. So I'll say a little bit about that towards the end. So the Canadian Network of Scientific Platforms is a group that was formed to bring people together from across Canada who are running scientific infrastructure platforms. And some of them are bioimaging. There's also platforms in flow cytometry, materials science, so nanofab facilities. There's some facilities that offer more service-based, so things like viral vector cores or biobanks and data storage. And so it's not just bioimaging, it's really covering all the way from engineering to health sciences. And these platforms really act like a hub. They interact with academic researchers. They have the expertise for the scientific infrastructure. They often have corporate partners either using the infrastructure or they're partnering with the companies that manufacture the infrastructure. And they really focus on training the next generation of scientists and engineers. So like in the ABIF, we train about 200 to 250 people every year. So these platforms are really a hub where graduate students and postdocs are learning the latest technologies and how to apply them to their scientific questions. So the group is pan-Canadian. We have 194 scientific platforms. You'll see that we have a lot in Quebec, and this is because the group was founded in Montreal. And we're working on building up our membership in Ontario. And we have representation from all the provinces except PEI. And no territories yet, but I guess we can work on that. And we represent about $1 billion of research infrastructure spending. So between the Canada Foundation for Innovation, the provinces, and the corporate partners. And this list, I've mentioned a few of these already, is just the different types of platforms that we have. So the Canadian Network of Scientific Platforms is really focused on the overarching technologies, professional development for people working in platforms, trying to advocate to stakeholders and funders about the need for more funding for sustainability of these platforms and for career paths and so on for the people working in the platforms. But we wanted to also have more technology-based groups. So the Canada BioImaging is a technology node underneath the Canadian Network of Scientific Platforms. And we have another group in flow cytometry that will be joining as a node soon. And we've also been working with a national group that's working on the helium crisis. I don't know if people are aware, but helium is a byproduct of the petroleum industry, and it's a finite resource. It's not unlimited, and there's been a lot of issues because a lot of it comes out of Russia. So there's a lot of supply chain issues and just global shortages. So we've been working with them to come up with strategies to help solve that problem. And then BioImaging North America encompasses Canada, the US, and Mexico. So Canada BioImaging is part of that network as well. So right now, Canada BioImaging has 86 members from 28 facilities, 8 provinces, and 38 institutions. So the goal of the group is to bring together the community around bioimaging, to coordinate courses and workshops across the country. So we're not offering the same types of courses at the same time. We're working on job shadowing, technology training, facility management training, and a lot of that is being done with BioImaging North America. And then best practices, quality management, and so on is something we're working on with Core App. So BioImaging North America was founded after Canada BioImaging, and the idea was to bring the US and Mexico into the community and really try to get those groups across North America working together. The idea is to share collective expertise within the community. So if people have the similar technologies, we can get together around those technologies and solve common challenges. A focus on standards and quality control. Community-driven working groups. So I'll tell you about, there's 6 working groups right now. Professional development and education for the imaging scientists working in the platforms. And then BioImaging North America is really our partner with the different global groups. So the working groups, there's 6 working groups right now, and these are basically driven by the community and the needs of the community. So there's a communications group that has a monthly newsletter, posts on Twitter, keeps the website up to date. The training and education group that focuses a lot on bringing resources together. So there's a lot of resources on the website that were collected during COVID and now moving more towards in-person courses and workshops. There's a diversity and inclusion group, which is trying to raise awareness of the situation for underrepresented groups and ideas of how to improve that. Quality management, or quality control and data management, which I've been quite active with that group, bringing awareness around research data management and then also providing tools that people need for it. There's a corporate partners working group that really engages with mostly right now, we've been working mostly with the microscope and camera and accessory manufacturers. And the idea is to also build up our links with biotechnology and just understand their needs and what the community, kind of what can we do together as a community that's win-win for both groups. So for instance, last year, Zeiss ran a pilot project where some of the imaging scientists from more remote facilities that have a difficult time getting service visits were taken to the Zeiss headquarters and given some training on basic troubleshooting for confocal microscopes. And then the idea is they could get a discount on their service contract and do some of the troubleshooting and service themselves. And lastly, there's imaging informatics, which is really trying to work on customized image analysis workflows, streamlining the different tools that are available to the community and so on. So some of the activities, I won't go through all of these, but there's meetings and workshops. They just had our first in-person meeting at Woods Hole in October. The Metrology Suitcase, I'll say a little bit more about, but this is, the idea here is to send the tools and the protocols around to different groups so they can quality manage their microscopes. And one of the most interesting thing that's come out of the group so far is this microscopy database. And what this is a centralized database where people can post training and education resources, workshops, courses, jobs. And the idea is that we have a common database for everybody globally, and that everybody will put their data into that resource. And then each member in the network or any network around the world can pull out the data they're interested in. So we just connected with this on the Canada BioImaging website, and now our events and our job pages are updating automatically. So it basically has a connection directly to the database. And if you go in and put a new job posting in there, it will show up on the Canada BioImaging website once it's uploaded to the database. So it was a lot of work there were a lot of communities around the world who were doing something similar, but the BINA administrative team really went around and talked to all the different groups and understood what people were doing. And I think it was a really good example of how these networks can work together to streamline things. So now it's kind of a one-stop shop but they weren't trying to replace like the Royal Microscopy Society website or the Global BioImaging website. So Global BioImaging still has their website, but they can pull their job ads up from the database. So it was a really nice project that's really just getting launched worldwide now. So the Metrology Suitcase program is, the idea here is to send the tools around as well as the protocols and information that people need to quality manage their microscopes. And this is modeled after a program that started in France. And the idea was that not everybody might have these tools in their facility, and they may not know how to use them well. So right now there's just 2 tests. We have what's called a PSF check slide and a tetraspect slide where there's fluorescent beads or fluorescent patterns on the slides, and you can measure the resolution of the microscope or the microscope settings with a specific lens. And then there's a power meter from Thor Labs that has a microscope slide shown here that can go on the stage, and you can measure the stability of your light source. And you can also measure the power of the light source. And these, the kit is meant to move around between different laboratories. And
Blair, we're not getting any audio. I don't know if you can hear us.
Not sure. Did it just stopped?
Okay.
How about now?
Yep.
Okay, I just changed the settings, so okay. So did it just drop off?
Yep.
Okay. So so right now there's 3 kits. There's 2 in the US, 1 in the Midwest, and 1 on the West Coast. And there's 1 in Canada. Is it okay?
There we go.
You're okay?
Yep.
Okay. So there's one unit in the Midwest and 1 on the West Coast of the US, and 1 unit in Montreal actually at the CR CHUM. And the idea is they'll quality check their microscopes at the CR CHUM and then share the kit around Montreal. And then the kit could be shipped to Toronto and shared around to some facilities in Toronto. And the data is going into a centralized database, which is being managed by Core Apolini. So we're thinking over time, if we get enough data collected with the same tools, that we can really start to mine that data and understand sort of maybe the common things that might go wrong with the microscopes, or maybe there's a certain brand of laser that's not working well, or in turn, maybe there's a laser that works really well and is always really stable. So that's the BINA initiative. So I'm going to move now on to global bioimaging. So chronologically, Global BioImaging was the first one that was formed, and then Canada BioImaging and BioImaging North America came later. And Global BioImaging was formed in 2016 with funding from the Horizon 2020 fund from the European Union. And it's really an international network of bioimaging facilities and communities. So Canada BioImaging and BioImaging North America are both part of Global BioImaging. And in October, there was a conference, the International Research Infrastructure Conference, or ICRI, was in the Czech Republic. And they asked the different groups to publish a video that would be shown to the delegates at the conference. So I thought as a starting point, I would show the video, and you'll see, get a bit of a snapshot of what global bioimaging is. So Phil, I don't know if you need to adjust the sound, but it's the video's coming now.
Understanding life, imaging. From a single cell to complex ecosystems, imaging technologies are crucial. In Brazil, scientists use microscopes to study tropical diseases and evaluate the influence of climate change. Researchers in Israel employ underwater microscopes to observe life beneath the sea. Imaging technologies also help us understand the human body and improve diagnosis and treatment. To solve humanity's greatest challenges, imaging scientists and infrastructures must be connected.
Global bioimaging is bringing together people, the communities in imaging. The inspirational stories
of where people come from, and that there's so much more than just a day-to-day life in a microscopy lab. There's a world out there that we are making an impact in, and we should see that bigger picture.
So we're building the Latin America BioImaging Network, and Global BioImaging has allowed us to have a stage to connect with the rest of the world.
The impact is immense, I would say, because it was through the Global BioImaging that I come into contact with a lot of absolutely fantastic and vibrant imaging communities.
Our mission is to promote and connect imaging scientists and imaging research infrastructures from around the globe. One way to be part of it is to join our working groups. So there are several with different topics, and anyone can join.
It's togetherness.
Exchange.
Cooperation.
Connection.
Community.
Inspiring.
So one of the goals of that community is really to raise awareness about bioimaging communities and imaging facilities, and most importantly, as they mentioned, the people, the imaging scientists who are working in imaging facilities and supporting the research. And so that video is part of a number of different initiatives to just raise awareness and demonstrate the impact that imaging science and imaging scientists have on science and research. So this map shows, again, how global things are. You saw the countries popping up in the video as well. You can see Canada BioImaging and BioImaging North America here, and Latin America and the African BioImaging Consortium are the most recent members. And there's emerging partners in Chile, other parts of Africa, Armenia, and South Korea. This is a photo from the last meeting of Global BioImaging, the exchange of experience in Montevideo, Uruguay, which was done in combination with the Latin American bioimaging community. And these meetings are really about exchanging experience. So the theme this year was all about how to measure and demonstrate impact. So the working groups are similar to BINAR, kind of the main component, and their image data management. And this group published a paper last year on open image data formats and repositories. There's a group on career development for imaging core facility staff, and they're working on an article which will focus on the presenting an ideal career path for people working in imaging facilities, aimed at institutions and funders to show the importance of having clear career paths for people working in platforms who are trained experts that really are required to make this infrastructure work. Increasing the involvement of the biomedical community, so they're trying to build up that group. And then societal impact of imaging infrastructure. So we published this document, I think it was in 2020, on an international recommendation on how to measure the impact of imaging core facilities. There's a training and education group and a quality management group. And I'm realizing the time is going fast, so I might speed up a little bit and maybe provide a little bit less detail. So another document that came out of this group was the added value of the open access imaging core facilities. And this is a really interesting document in that they broke it down into the different stakeholders. So what's the advantage for publishers? What's the advantage for microscope manufacturers? What's the advantage for the research community? And it's a very light document in that it's just a few pages and pulls in the highlights. The training initiatives are a few different formats. So there's an online training resource. There's an international job shadowing program where people can travel between facilities to learn either a new technology or facility management experience. And then there's courses and workshops for training facility staff. So the last group I want to talk about is the Core Apolini group. And this is a figure from a paper that was published in 2021 in Nature Methods, introducing the community to the global community. And this group came together really quickly during the pandemic. It was started in April 2020, and at that time, a lot of people were in lockdown. So I think the timing had a little bit to do with how quickly the group grew. But there's 462 members from more than 35 countries. And as you'll see from this figure, there's a huge involvement from many different groups, not just academia. So the academic groups shown here are the networks, so similar to Canada BioImaging, the different networks around the globe. The manufacturers, we've been really impressed at the number of companies that have gotten involved with the group and are really wanting to work with us to improve quality management in microscopy. The standards organizations, so the ISO, the National Institute of Standards and Technology in the US, and another example is the German Standards Institute, and then some sort of overarching groups such as Euro BioImaging, Global BioImaging. And it really is making up a good cross-section of the community. So it's broken up into working groups as well. Some of them are really specific. So work group 1 to 6 are focusing on very specific measurements of quality on a microscope. So the illumination power and stability, the lateral and axial resolution using microspheres, uniformity of the field, reproducibility of the stage. And then there's a few groups that are on more overarching, like the metadata. So really working on what information needs to be provided for research data management. And then some of the later groups that formed are image quality, publication standards. I'm involved with the phototoxicity group as well, trying to work on recommendations for accounting for and reporting phototoxicity. And I'll just mention for the publication standards, it's been quite interesting, sort of in relation to that Nature Methods article. The Nature Publishing Group has reached out to the COREP community to build a checklist for microscopy for all of their journals. So this is a checklist that the authors would go through to make sure they provided all the appropriate information. So they've been working on publishing protocols, and especially those first 6 groups I mentioned. So there's a website called Protocols.io that's an open access online journal. And these are 2 of the protocols that have come out of the working groups. And it's a nice platform in that you can work back and forth with your colleagues privately and then publish the protocol when you're ready. And once it's published, people can comment on it, you can make changes, and then you can publish a revision, and the historical documents are maintained. So I'd mentioned the article in Nature. They actually identified reporting and reproducibility in microscopy as a priority in 2021 and had this whole special issue with a number of different articles on bringing awareness to quality management and also presenting tools. And this paper here was headed by Joel Ryan, who's in the room, where he built on some code that was developed at, I'm going to forget now, Mark Sanders' lab. Sorry, and was MethodsJ, so we cleverly called it MethodsJ too. And it's a tool I'll say a little bit more about, but to pull together information for your methods section of your papers. There were several other papers, the COREP paper itself, a paper on best practices and reporting of microscopy information, and so on. So one of the tools is Micrometa App, which is collecting what's called metadata. So metadata is data about other data. And it's a software tool to help you collect information about all the different aspects of the microscope and all the different components, so that when you publish an image, the person can go back and see how that image was generated. And that tool can then be combined with the MethodsJ tool and other tools like from the open microscopy environment to collect all the information you need to publish and report on your images. So this is a more in-depth snapshot of the Micrometa app. You can see the physical microscope here, and then within the app, you can go in and put all the details about the objective lenses and the filters. And once you've done that, then anybody who publishes with that microscope can just refer to this file, and the person who's reusing or sharing the image will know all the different properties that were, all the different components that were involved in collecting that image. And so this can then be connected with in ImageJ with the image metadata coming from the raw image data. So the maybe the pixel size or the exposure time and things that are included in your images. And then you can take the Micrometa app information and you can combine those two. And in the MethodsJ script, you can go through step by step and choose the different components that you used in your experiment. And then it generates a first pass at a methods text that can be basically copied and pasted into your paper. And we can also add an acknowledgement section if you've been using, if the microscope is in an imaging facility, for instance, you can see there's you probably can't read the text, but there's an acknowledgement there for the ABIF. So this just shows the workflow, which is also available in the paper. And this basically just shows that, you know, Canada BioImaging, the ABIF, McGill, we're part of this really big ecosystem of international networks. We're part of an international network of networks with a lot of different activities going on. And maybe I'll just point out 3 things. So we're doing a Montreal Light Microscopy fundamentals course here in Montreal in February and March over the winter break. We're also working on a national cloud-based image data resource to store, manipulate, and share images. And there's a super resolution course that's being offered as part of Canada BioImaging at the University of Calgary from May 8th to 12th, 2023. So if you want any more information about those projects or those events, feel free to let me know. And I think that's my last slide. So happy to take any questions.
Can I ask a question?
Please go ahead.
So you mentioned at the beginning, you know, this is the community, and I believe you encouraged us to join. And yes, I'm interested in maybe, you know, selecting one of these organizations and joining and based on my expertise and contributing. So what should I do? What should I do?
I think you could reach out to me directly, and if you have specific topics or things that you'd like to be involved with, like if you're interested in standards, it would be the core app group. If you were more interested in, you know, teaching and training and education, I would probably say the BioImaging North America or Canada BioImaging. So we could dialogue back and forth a bit and see what's the best fit.
Thank you.
Thank you.
Claire, maybe building on that question, I guess we have a room full-ish of students and postdocs. So where would they start, or what would you recommend for them in terms of resources that are available, or things that they could do to be involved?
So just for the Zoom group, the question was, how could the students or postdocs get involved with the networks? So I think it's a similar answer as well. I mean, it depends what they're interested in. And I know if people want to discuss with Natalie, I'm not sure if you're going to the Wine and Cheese, Natalie, but she's involved in the diversity and inclusion group with BioImaging North America. And I didn't talk about, but we've just recently set up some user groups around different technologies. So Natalie and I started an expansion microscopy user group, which now has over 100 members from 23 countries. And it formed so quickly because we had this international network to rely on. And it's really geared around helping people do expansion microscopy. So it's really helping for Natalie getting started on her master's project. And then hopefully we'll help others, you know, around the globe do that more quickly as well. We're starting a second one on lattice light sheet since we've just gotten a system at McGill. And then, you know, if people are interested in training and education, or they're interested in you know, corporate outreach, maybe they want to get to know, you know, careers in the microscopy companies, they could join the corporate outreach community and get to you know, understand their needs and sort of how they're engaged with the community. So I think we're actually with BINA, we're really we focused originally of just building the community around core facilities, but our one of our goals for 2023 is to get more students and postdocs involved.
So A small question. You mentioned the second light sheet microscope. Is that in ABIF, and is that for hydrophilic medium, or can we do iDisco Plus in it?
So it's a lattice light sheet, so it's a high resolution system, and it's designed for live imaging.
Right.
So it's meant for imaging live tissue or live cells. Yeah. I did want to just come back when I think about the your question, Joel, is the other really interesting thing is these job shadows and exchange of experience. So if there are students who are interested in microscopy technology and they'd like to apply it in their project, but they don't know where to start, they can apply for funding from Global BioImaging or BioImaging North America to travel to a research lab or travel to a facility and learn that technique. And it could be, you know, a single visit, it could be 2 short visits, it could be a month-long visit. So those programs were kind of stalled during the pandemic, but are reopened now. So I'd be happy if people have certain areas they like to get more experience in to put them in touch with the right people for that. Any other questions?
I think that might be it.
All right, I'm happy to share the slides because I know I gave a lot of information in a very quick time, and we'll put the recording on the ABIF channel for YouTube, and there is wine and cheese in Bellini room 132 for anybody who's available, and anybody on Zoom, if you want to come over to Bellini, you're welcome to do that as well. And I guess our next session will be in January or February, so we'll keep you posted. If you have topics you'd like to hear about, feel free to let us know as well.
Thanks so much, Claire.
Thank you, Claire.
Thanks, everybody.
Thank you.
Thanks.